Hi, AICHE. Thank you so much for attending our presentation. My name is Brandy Brown, and I'm a doctoral candidate in bioengineering at the University of Nebraska-Lincoln. I'm pretty excited to share with you guys our study of taking this very metabolically robust microorganism and engineering it to produce bioplastics from lignin breakdown products. So in our review, I'll talk about why this organism, why did we choose it, and why lignin in particular. The growth analysis study that really provided us a good foundation of its catabolism of lignin breakdown products. Then we'll go into bioplastics and how we were able to engineer it to produce bioplastics from lignin breakdown products for the first time. And then our optimization of a bioplastic quantification method using flow cytometry. So why lignin and why Rhodopseudomonas palustris, or R. palustris? So typically when people think of agricultural waste, they think of the sugars that come from the cellulose that have been able to be turned into a wide variety of high-value products like chemicals and fuels. However, lignocellulosic biomass also has lignin, hence the name. Lignin can comprise up to about 26 to 30% of the lignocellulosic biomass itself, and so it's a considerably large portion of the biomass. However, you can see here that it's a pretty complex structure of phenolic compounds, and unfortunately, therefore, it's just more commonly burned for energy because it's difficult to break down and turn into products. However, if we could find, create, engineer a microbe that's able to utilize this lignin, break it down, catabolize it, we would be able to turn this large portion of agricultural waste into high-value products as well. Which is why we focus on R. palustris for this study. And so I'm a part of this Dr. Saha's Systems and Synthetic Biology lab at the University of Nebraska-Lincoln. And one of our main goals is to engineer R. palustris as a biotechnology chassis. This is one of the main efforts that fits into this overarching goal to bring that into fruition. And so we choose R. palustris for the studies because it's a very metabolically robust organism. It can take a wide variety of waste products. In particular for this study is the aromatic compounds, which would be the lignin breakdown products, and also turn it into a wide variety of value-added products like hydrogen or bioplastics is what we would focus on in this case. So before we went forward, we wanted to conduct a really in-depth analysis of R. palustris lignin catabolism. And so we did this by analyzing its growth and for a variety of different lignin breakdown products, or the monomers that lignin breaks down into, under aerobic and anaerobic conditions. And so here are some highlighted examples for aerobic. And you can see that R. palustris grows on a wide variety of the lignin breakdown products, as well as Kraft lignin supplemented with acetate. And we chose Kraft lignin because it's one of the major contributors to the most lignin produced worldwide, and so we thought it would be a good proof of concept for this study. And in general, this was the first time that this analysis had been conducted for this microbe under aerobic conditions for the vast majority of these lignin breakdown products. So also under anaerobic conditions, it was able to grow on all of our monomers that we analyzed. But these are some highlighted examples. In particular, p-coumarate and coniferyl alcohol grew to a significantly higher absorbance, 2 to 3 times that of acetate alone, and without being required to be supplemented with acetate. Kraft lignin, and compared to acetate, so it's this white line here compared to the green line, the Kraft lignin supplemented with acetate doubled the biomass compared to acetate alone. And so we are really, really intrigued by this, and this is really fascinating, and so our study of why this is the case is still ongoing. So this is the kind of summary table of all the lignin breakdown products and all the conditions that we subjected R. palustris to. And so this is just highlighted to see that the vast majority of them had not been reported previously. And so overall, this just gave us a really good foundation moving forward for the catabolism and the metabolic pathways that R. palustris is able to use. So now that we had this foundation, we wanted to produce bioplastics from lignin breakdown products for the first time. And the reason why this is so important is because there are 2 main reasons for increased bioplastic costs that are kind of inhibiting their widespread adoption compared to petroleum-based plastics. And these two reasons are high carbon costs and the cost of bioprocessing for extraction, essentially. And so these two efforts, the bioplastics from lignin breakdown products, aims at helping produce these bioplastics from a more renewable source and possibly a more cost-effective source to help potentially foster more widespread adoption. And then the last one is our high throughput method, which helps quantify the bioplastics. So this is an experimental workflow of how we would actually produce the bioplastics in the cell. The concept is we would use the lignin breakdown products as a carbon source for fermentation. We would then promote some sort of stress condition for the microbe so that it would be more willing or produce more of these granules inside of the cell, inside of the cytoplasm, which are these white inclusions that you see. And then we would extract it via chemical lysis or two-phase extraction, conventional methanolysis, if you will, and then quantify it with gas chromatography. So before going forward, since this is the first time this study had been conducted, or the first time we were producing bioplastics from lignin breakdown products from this microbe, we needed to analyze which condition or which stress condition would be optimal to produce the most bioplastic titer. And so as you see here, this is a comparison between nitrogen starvation, phosphorus starvation, and a combination of both nitrogen and phosphorus starvation. And so this enabled us to conclude that nitrogen starvation is the optimal condition that would produce the most bioplastic titer for this microbe on lignin breakdown products, and therefore we use this starvation condition going forward. So in order to produce bioplastics using nitrogen starvation, like we just saw, we also chose the two lignin breakdown products that reached a significantly higher absorbance in anaerobic conditions that did not require supplementation with acetate, and if you remember, that was p-coumarate and coniferyl alcohol. So using these lignin breakdown products and using nitrogen starvation, we were able to produce a bioplastic copolymer for the first time on lignin breakdown products from this microbe. This was also the first time that we were able to show that this microbe makes this copolymer called poly-3-hydroxybutyrate-co-hydroxyvalerate, or PHBV as is commonly known. And the reason why this matters is because PHB, just with the 3-hydroxybutyrate repeats basically as a polymer, is more brittle. And adding this 3-hydroxyvalerate component really boosts its thermomechanical properties and therefore widens its applicability. So we then wanted to get an idea of how the granules are forming inside of the cytoplasm. So we used transmission electron microscopy to accomplish this. And so this is a comparison in panel A to acetate, which had almost little to no bioplastic production, and panel B and C with p-coumarate and coniferyl alcohol, the lignin breakdown products. And you can see there's a stark difference, and the white inclusions inside of the cell, those are the bioplastic granules. And what's even more interesting is that this microbe produces one large granule inside of the cytoplasm in comparison to, let's say, a model organism like Cupriavitis necator that produces several smaller granules inside of the cytoplasm. And so this is really fascinating, and it gives us maybe options going forward so that we can optimize or engineer this microbe for more enhanced or maximum bioplastic production. So now that we had the growth analyses, a good foundation of catabolism, we produced bioplastics from lignin breakdown products for the first time. We wanted to thwart the other leading costs of increased bioplastic production costs, and that's the quantification. And so we looked at optimizing a study that had already been reported from University of Nebraska-Lincoln previously. And it was basically a study that utilized flow cytometry to quantify polyhydroxybutyrate in Cupriavitis necator, the model organism that I was just mentioning. And this is the schematic, or basically the experimental workflow for this study. We would produce the bioplastics similar as I was discussing previously with using the lignin breakdown products as a carbon source. We then would subject it to a permeability assay with the hypothesis that this permeabilizer solutions will enable these fluorescent dyes to penetrate the cells, and therefore we'd be able to quantify the bioplastics inside of the cell, as well as get a good bioplastic production on a per cell count basis. And so previous studies, while they rendered good linear fitness for bioplastic titers compared to these fluorescent dyes, the fluorescence of the dyes, the cell count did not render such a good linear regression. And so we wanted to optimize this so that way we could get bioplastic production on a per cell count basis, basically. So these are the results of our permeability assay, and overall, the major takeaway is that we found that 50% ethanol and 3 microliters volumes of stain for either Nile red or Bo-Dip-B were ideal. And I should mention that Nile red is more conventional, and it's been used, I guess, more widely used, whereas Bo-Dip-B, this is the first time that this fluorescent stain has been used with our palustris in general as well. And so this really gives us a good idea on the comparison of these fluorescent dyes and the applicability, the wider applicability of this method as well. So using those conditions, the permeabilizer conditions, these are the results of our linear regressions between the bioplastic titers as well as the cell count. So you can see for both dyes, both for Nile red and Bo-Dip-B fluorescent stains, that yielded a very positive linear fitness for both the bioplastic production as well as on a per cell count basis. And so overall, this helps us be able to quantify bioplastics quicker without having to go through the conventional acidic methanolysis, which takes toxic chemicals, takes several hours, and gas chromatography. This, you can just stain the cells, subject them to flow cytometry, and have a very good, quick and reliable way to quantify your bioplastics on a per cell count basis. So in summary, we utilize this very metabolically diverse microbe called R. palustris to catabolize lignin, and we produced an in-depth growth analysis study on a variety of lignin breakdown products as well as Kraft lignin for the first time. We produced bioplastics on these several lignin breakdown products for the first time, and we also showed that this microbe can produce this copolymer for the first time. And lastly, we optimized a high throughput quantification method of these bioplastics using flow cytometry. I would like to take the time to thank to our funding sources, the Corn Board, University of Nebraska-Lincoln, Nebraska Center for Energy Sciences Research, and my colleagues for always laughing at my corny jokes. And of course, I'd like to thank you, and I really appreciate you taking the time to come to our presentation. I look forward to seeing and hearing from you at our Q&A session, and I look forward to attending all of your presentations. Thank you so much.